Now, let's see the process of meiosis. I told you in the previous videos that meiosis is a type of reductional division and mitosis is an equational division. Now, we can divide this meiosis in two phases, that is meiosis 1 and meiosis 2. Actually, this meiosis 1 is a reductional division and meiosis 2 is almost same as mitosis, and that's why we can also call it as equational division. Now, we can divide this meiosis 1 into the karyokinesis and cytokinesis as we were doing in the mitosis. There also we have seen the phases of the mitosis, that is karyokinesis and cytokinesis. Here also we will see the meiosis karyokinesis 1 and cytokinesis 1. When we come to the meiosis 2, then karyokinesis 2 and cytokinesis 2. Now children, this number is there just to avoid our confusion, because mitosis also consists of the karyokinesis and cytokinesis. Meiosis also consists of the cytokinesis and karyokinesis. So just to avoid all these confusion, in case of mitosis, we use only karyokinesis and cytokinesis. In case of meiosis 1, we use karyokinesis 1 and cytokinesis 1. And in case of meiosis 2, we use karyokinesis 2 and cytokinesis 2. So, if a question will come to you, that is cytokinesis 2 is of which type of division, then automatically you will say that it is of meiosis 2, because from the 2 it will be easy for you to consider that it is from the meiosis 2. Now, this also we have studied earlier that karyokinesis can be divided into 4 phases, that is prophase, metaphase, anaphase and telophase. In the same way, in case of meiosis 1, we will give prophase 1, metaphase 1, anaphase 1 and telophase 1. Then cytokinesis. After this, we will enter into the meiosis 2 stage. There, karyokinesis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, which is followed by the cytokinesis 2. Now, this much of detail is not there in the course, but the summary of the meiosis is there, and that I am going to give you now. During meiosis, a cell undergo 2 division, one after another. And as I told you that it occurs in the sex cell. So, let me draw one cell. Clear? And suppose it is consisting of a pair of chromosomes. Let me draw this. One more pair. Now children, I told you that we all have 23 pair of chromosomes. Clear? But just for our convenience, I am taking only 2 pair of chromosome. So we can write it as it is 2n. Okay? Now see what happens before any cell enters into the meiosis. It first undergo for the interphase. If you remember, in case of mitosis also, we have seen that before we start the prophase, metaphase and all other phases, our cell was going in a most active period that was called as the interphase. So the same thing happens in this also. A sex cell will go into the interphase. Now in case of interphase, synthesis phase is there, where the DNA replication occurs. So what will happen in this case? Before entering into the prophase 1, the cell will undergo DNA replication, that is, it will go in the interphase. And if it will go in the interphase, it will make one more copy. Okay, now suppose I am drawing one more copy. This is only the copy. Okay, don't get confused. It is only the replicated copy of the DNA. After making the DNA replication, now the cell has entered into the prophase 1 stage. Now see what happens. I told you that all 46 chromosomes are there and they will search for their own pair and they will pair with them. That is 23 pair will come close together. Now, if I have taken 4 chromosomes here, then the 2 will come close together, pairing will occur, and if pairing is there, then we can call it as a homologous chromosome. This is again one more piece of homologous chromosome. Understood? Now, these homologous chromosome, they are like this. Clear? If we move to the next phases of the meiosis 1, then these chromosomes will lie over one another, and if they will lie over one another, then they will cross each other. Okay? And if they will cross each other, then their genetic material will exchange. And this is called as the crossing over. And because of this crossing over, recombination occurs. Now see the same thing what I have said through the diagram. See, suppose the cell is entering into the next phase of meiosis, and suppose if I draw the chromosome like this, okay, this is one more chromosome, and I told you that it is replicated piece, so I am drawing the replicated piece also. This is for this one. See, clear? Now, this is one homologous pair. This is another homologous pair. Now what will happen? This will come close together. This will move together. During this, what they will do? They will overlap with each other. Clear? Suppose I am drawing it like this. They all will overlap with each other. The same thing will happen here also. This will overlap with this, and this will overlap with this. Now see, this is the overlapping point, and this is called as chiasmata. And the process is called as crossing over. Understood? Now see once again, we have a pair of chromosome. They come close together. After that, if they will enter into the separation stage, then randomly they will separate. Now, if they all have come close together, it means their chromatids, they will overlap each other. And now when they will get separated, then the segment will exchange. Now this segment will exchange. Now see this in the next stage. Suppose I am drawing it here. This one is the blue one. Okay. After crossing over, now this has been crossed over. So what has happened? This portion has become darkened and this one has become light. In the same way, this portion has become darkened. Same thing with red one also. This is the normal chromosome and this is the crossed chromosome. Okay. Now this is called as the crossing over. After this, what they will do? They will undergo into the last stage of meiosis and they all will get separated. Now see how they will get separated. Suppose if I am drawing it like this, then this chromosome will go in this column Okay, and this chromosome will come to this side. Now you just see, here we have taken 2n. Here the crossing has occurred in 2n, and while separation, the genetic segment has been separated between the two. That is, the intermixing of the maternal and paternal genes has taken place due to crossing over, and after that they all get separated. Now see, this is N and this is N. This whole step is called as meiosis 1. Now, this N will enter into the next step, that is meiosis 2. You see once again, this is a pair of chromosome. At first, replication has already taken place. Now, after this, the paternal and maternal chromosome, that is the different chromosome, they come close together. There is a crossing over. After crossing over, genetic material has been exchanged and then they divided into the two. Now half chromosome is here and half chromosome is here. It forms haploid now. Clear? And here it was diploid as I told you. Now we will enter into the second phase, that is meiosis 2. Where to draw? I am drawing it here only, children. Okay, this stage. This I have already drawn. That is, I have drawn this with blue. Chromosome, chromosome. Okay, this is again chromosome. Chromosome with its replicated. Don't get confused in this and this that it is a 2. Okay, it is only the replicate piece that has already done in the interphase. Now, this replicated piece will get separated as we have already studied in the mitosis. That is, same as in mitosis, we have seen that replicated chromosomes, they form chromatin material, they condense to form chromosome, then they form spindle fiber, then they stretched by the spindle fibers and they shift towards the pole. And after that, there was a cytokinesis and then we got the two different cells. The same thing will be there. See, this will get separated. The copy will get separated. Okay. Now, see how the copy will get separated. This is one piece. This is another piece. But here crossing over. Genetic exchange we have to show na. Genetic exchange. This is one piece. And this is one more. Okay. This is also N. Now it will form 4 daughter cell. Now this stage is called as the meiosis 2, where already N has been divided. Now this is equational division because N number has not reduced to N by 2. Only whatever was the copy, that copy was separated into the different daughter cells. So, this is all about the process of meiosis. Now, once again I have to revise the things. You see, chromosome duplication, then chromosome become half. While making them half, there was crossing over. After crossing over, we got the new type of this chromosome. And now student, keep this thing in mind. If variation is there, okay, then that is just because of the crossing over. And that's why meiosis is also responsible for the evolution. Because during meiosis, we got the variation because of the change of the genetic material, exchange of the genetic material. Now after this, we'll get the 2 haploid, then there will be the equational division. They all will get separated and then they form the 4 haploid daughter cells. So this is all about the meiosis. Now let's see what is the significance of the meiosis. I told you, now the significance is clear to you all. That is, we can see variations. Okay. So number 1, significance. This meiosis maintains the number of chromosomes in organism. Okay, now see how it maintains the number of chromosome. As I told you, there is a chromosome with one. Now suppose if it is sperm, same thing has been made in the ovum also, like this. Now if they will fuse, that is after fertilization, if they will fuse, they will form the zygote, and then now it will become 2n. Clear? So this haploid will again become diploid, and that's why the number is same, number is constant. If it is only 23, it is only 23, it will be 23 pair. This is the significance of the meiosis, that it maintains the number of chromosome in the organism. Now, next significance, this meiosis occurs the exchange of maternal and paternal characters. Clear? How it maintains the maternal and paternal character? By the exchange of chromosomes, and we all know that chromosome contains gene. So by the exchange of gene segment or by the exchange of chromosome piece, the exchange of maternal and paternal characters occur, and that is because of the crossing over. Now, I think so this is all about this one. One more we can write, that it leads to the variation, okay, which finally causes evolution. This is all about meiosis. Now children, I think so we have completed chapter number 1. Okay, now in your exam you have to prepare all the types of or different phases of the mitosis and general rules of the meiosis. Along with this, the definitions that I have told you in the previous videos that we all have to do. After this, I'll give you 2 or 3 questions also, and I hope that you people will note down those questions and then you will solve it. Clear? Okay, thank you.